Now we have seen in second part of hyphenated techniques. As per our syllabus, three hyphenated techniques are described. First GCMS, second LCMS, and third LCNMR. Now, firstly we have seen GCMS, hyphenated technique. GC means gas chromatography is able to separate the volatile and semi-volatile compounds, but it unable to identify them, whereas mass spectroscopy, which is short formally called as MS, can identify the compound by giving its structural information at molecular level, but it unable to separate them. Therefore, the combination of these two techniques took place shortly after the development of GC, means gas chromatography. So GCMS is called as gas chromatography mass spectroscopy. GCMS was the first technique to be hyphenated, and this technique can confirm the organic, semi-volatile and volatile compounds and the residual solvents with great resolution. For the analysis of the compound by GCMS, the compound should possess the property such as volatility and thermal stability. Now we have seen instrumentation and working of GCMS. Vaporized analyte, when carried through the gas chromatographic column with the help of heated carrier gas, the separation occurs in the column only. Carrier can also be called as mobile phase, for example helium. Distinguishable interactions of analyte between mobile phase and stationary phase lead to separation of the compounds. The separation of the analyte is also depend on the column's dimensions, means length, diameter, film thickness. Then type of carrier gas, column temperature, gradient, and the properties of the stationary phase. The sample travel through the length of column. The difference in the boiling point and other chemical properties lead to separation of the components of the mixture. The components will be having differences in elution time and retention time due to their different absorption or difference in the partition between mobile phase and the stationary phase respectively. Then the separated components of the mixture will enter into the mass spectroscopy, means MS, through an interphase. This is followed by ionization. Mass analysis and detection of mass to charge ratios of ions generated from each analyte by the mass spectrometer. An interface like effusion separator, jet oblique orifice separator, and membrane separator can be used to connect GC with MS. The process of ionization not only ionize the molecule but also break the molecule into the fragments and detect these fragments with the help of electron impact ionization and chemical ionization. The molecular ion of analyte form a fingerprint spectrum which is different from other analyte. GCMS is important tool in analytical chemistry because these techniques accurately separate. Applications of GCMS. First, quantification of pollutants in drinking and wastewater using official United States Environmental Protection Agency methods. Second application, quantitation of drug in metabolites and urine is done for the pharmacological and forensic use. Third application, identification of unknown organic compounds in hazardous waste dumps and reaction products by synthetic organic chemistry. And fourth application, used for drug analysis, pesticide and herbicide detection. Now we have seen second Hyphenated technique, that is LCMS. LCMS means liquid chromatography mass spectrometry. So this is the technique which performs separation by liquid chromatography and mass analysis with the help of the mass spectrometry. With the help of HPLC, impurities and degradation products can be separated, and mass spectrometry allows us to obtain the molecular weight and identification of the same. LCMS is highly selective and sensitive technique. LCMS leads to detection and identification of chemicals in presence of other chemicals. Therefore, it is called as specific. The flow rate of HPLC is around 1 ml per minute, which is difficult to accommodate in mass spectrometry. Vacuum system is also the diluent which is used has to be vaporized, which lead to damage of the thermally liable compounds by excessive heating. By hyphenation of these two techniques, capabilities of both the techniques were improved. Instrumentation and working of LCMS. The LCMS instrument can be interfaced by electrospray, particle beam, thermospray. Electrospray is most widely used interface. The spray needle is used as bridge to connect the liquid chromatography with that of the mass, but the separate emitter is flexible as well as convenient. LCMS is mainly separated into three parts: chromatography, interface, and spectrometry. In liquid chromatography, separation is performed, which is detected with the help of photodiode array, ultraviolet, fluorescent detectors. These separated components then transferred to the interface. In interface, the liquid is volatilized and transferred to the MS. With the help of various ionization techniques, the compound is ionized and then it is analyzed by mass analyzers. Various mass analyzers are used, for example, quadrupole ion traps, time to flight reflections, and ion cyclotron resonance. Mass analyzers in this technique liquid chromatography separate, due to which clear. Applications of LCMS. First application, LCMS used to detect compounds from polyaromatic means non-polar to peptide and proteins. Second application, LCMS used for compounds identification and purity. Third application, it is used for determination of pesticides, herbicides, and organic pollutant for environmental monitoring. And fourth application, proteome analysis is done by this technique. Now we have seen third hyphenated technique, that is LC-NMR. LC means liquid chromatography and NMR means nuclear magnetic resonance spectroscopy. So LC-NMR is the hyphenated technique in which HPLC is combined with the NMR. This technique is widely used for analysis of complex mixtures which contain unknown impurities, natural products, and synthetic polymers. In LC-NMR, LC does the separation and NMR does the identification of separated components. Now we have seen instrumentation and working of LC-NMR. LC-NMR is the sensitive method because the sensitivity of the NMR can be increased by use of highly magnetic field magnets and highly sensitive probes, and maturation of peripheral technologies such as solvent elimination technology and automatic measurement software suitable for multi-component analysis. Stronger the external magnetic field is, the higher the sensitivity. The improvement in sensitivity has made for a large reduction in measurement time. The compounds showing complex spectra can also be easily analyzed with the help of increase in magnetic field, and which will improve the signal resolution. Applications of LCNMR. First application, identification of drug degradation products. Second application, low level impurities can be isolated and identified. And third application, this technique is used for tracking pesticides, herbicides and organic pollutants for environmental